Imagine you're running a clinical trial, right? You have this highly anticipated leukemia drug, and the drug is actually working.
Right, it's doing its job.
Yeah, but suddenly, like, 20% of your patients experience this terrifying side effect. Their platelet counts just completely crash.
Which is incredibly dangerous.
Exactly. And in the medical world, I mean, most researchers would just flag that as an adverse event, slap a black box warning on it, and move on.
Yeah, that's the standard protocol.
But what if that glaring flaw, that bug in the drug's design was actually the exact hidden feature you needed to cure a completely different, completely fatal bone marrow disease?
It's just a massive paradigm shift in how we think about drug development. You're looking at this severe side effect and asking, well, who could actually benefit from this specific kind of cellular destruction?
Right. And that is exactly the focus of our deep dive today. We are unpacking this fascinating 2026 paper from the journal Cancer Cell. It's by Wen and colleagues.
A really brilliant piece of biological detective work.
It really is. The mission for us today is to understand exactly how this team of researchers did that. They reverse engineered a dangerous side effect to target a notoriously brutal blood cancer. So whether you're a medical professional or just, someone who loves a good scientific mystery, we're going to show you how they connected those dots.
And the disease they targeted is one that desperately needs new ideas. It's called primary myelofibrosis, or PMF.
Okay, let's break that down. What exactly are we dealing with here?
So PMF falls under this broader umbrella of diseases called myeloproliferative neoplasms, or MPNs. Basically, PMS is a cancer driven by genetic mutations. Usually we're talking about mutations in genes like JAK2 or MPL or CALR.
Right, those are the big three offenders.
Exactly. And these mutations create this state of what we call cytokine-independent signaling.
Which means what exactly?
Well, normally your cells wait for the body's chemical instructions, the cytokines, to tell them when to grow or divide. But with these mutations, the cells don't wait anymore. The gas pedal is basically just glued to the floor.
So they're just wildly overproducing blood cells.
Yeah. And clinically, I mean, it's devastating. For high-risk patients, the median survival is just a couple of years. The disease actually forces the body to start manufacturing blood outside of the bone marrow.
Like in the liver and the spleen.
Right, which causes massive swelling. But the really defining horror of PMF, the thing that makes it so lethal, is the fibrosis itself, the scarring.
Yeah, the bone marrow scarring. And reading through the paper, the mechanism behind that scarring is just fascinating. It traces back to a very specific type of cell called a megakaryocyte.
Oh, yeah. Megakaryocytes are key here.
Normally, they're essential, right? I like to think of them as these massive, giant cellular factories living in your bone marrow, and their sole job is to manufacture platelets for blood clotting.
That's a great analogy. But in PMF, those factories go rogue. They become what we call atypical megakaryocytes.
And it's not just that they're pumping out the wrong number of platelets, right?
No, the real issue is their exhaust. These rogue factories start leaking this toxic sludge, a cocktail of highly inflammatory cytokines, right into the marrow cavity. And the primary culprit in that sludge is one called TGF-beta.
Ah, TGF-beta. So it acts almost like a rogue foreman on a construction site. It essentially commands the surrounding tissue to just endlessly deposit collagen.
Precisely. Your bone marrow is supposed to be this soft, spongy, highly vascularized environment, right? It's the perfect nursery for generating new blood. But TGF-beta forces the tissue to lay down thick layers of fibrous tissue instead.
So the marrow literally turns into rigid concrete.
Exactly. The environment is completely destroyed.
Okay, but wait, let me push back here for a second. Because we do have treatments for PMF right now. I know a drug called ruxolitinib is prescribed for this. So why are these researchers digging for a totally new approach if we already have a targeted drug?
Well, because ruxolitinib, which is a JAK inhibitor, it has fundamental limitations. I mean, it acts as a signal jammer. And clinically, look, it's incredibly useful for managing symptoms. It drastically shrinks that enlarged spleen we talked about. It calms the systemic inflammation, and it definitely buys patients time. But it's essentially just a weed whacker.
Right. It trims the weeds down so the yard looks better, but the roots are still completely intact underground.
That's exactly it. It does not meaningfully reduce the mutant allele burden.
Meaning the actual population of the cancer cells.
Right. And more importantly, it doesn't clear the concrete. It does not reverse that bone marrow fibrosis. Plus, for the vast majority of patients, the drug just stops working after maybe 2 or 3 years. The disease is smart. It figures out a workaround.
Okay, so a weed whacker isn't going to cut it. We need a wrecking ball.
We do.
And this is where the cancer cell paper pivots to that leukemia trial I mentioned at the very start of the deep dive. The trial was called Augment 101.
Right. So Augment 101 was testing revumenib, which is actually an FDA-approved drug for heavily pretreated acute leukemias.
Specifically leukemias with KMT2A rearrangements or NPM1 mutations, right?
Yes. And for those specific leukemias, revumenib is an absolute powerhouse. It works incredibly well.
But then came that side effect. 15 to 20% of the patients developed grade 3 or grade 4 thrombocytopenia.
Which is a dangerously low drop in platelets.
Right. And that tells a very specific biological story if you're actually paying attention.
Exactly. The researchers saw this and connected the dots. If revumenib is wiping out platelets, the drug must be shutting down the megakaryocyte factories.
So their thought process was, well, if atypical megakaryocytes are the exact things leaking the toxic sludge in PMF, what if we use this leukemia drug to deliberately nuke the factories in PMF patients?
It's such an elegant leap of logic, but, a hypothesis is just a guess until you can actually map the cellular mechanics.
Right.
They had to prove exactly how revumenib was shutting down these factories at a microscopic level.
Okay, so let's do a quick recap before we dive into those mechanics. PMF is a deadly bone marrow scarring disease driven by rogue megakaryocyte factories. Current drugs fail to fix the root cause, but a leukemia drug's weird side effect offers this radical new clue.
Perfectly summarized.
So for you listening right now, I want you to think about your own life or your own work. Have you ever noticed a glaring flaw or a bug that actually turned out to be the exact feature you needed to solve a completely different problem? It's a really cool concept.
It happens more often than you'd think in science.
So let's look under the hood. What is this drug actually doing inside the cell?
Okay, so revumenib targets a specific protein called menin.
Menin, got it.
And menin doesn't operate alone. It functions in a complex with another protein called KMT2A. Together, they bind to the cell's DNA and physically enable the expression of specific target genes.
Okay, so let's unpack this with an analogy. If gene expression is like constructing a new skyscraper, KMT2A is the lead architect holding the blueprints.
I like where this is going.
But menin is the physical scaffolding. So without menin holding the construction workers up, it doesn't matter what the blueprints say. Construction halts. Revumenib essentially kicks away the scaffolding.
That is the perfect visualization.
Yeah.
By inhibiting menin, revumenib halts the gene expression that these rogue cells desperately rely on.
But I have to ask, menin is a fundamental scaffolding protein, right? Isn't it used by a massive variety of cells in the body?
It is.
So if you introduce a drug that just indiscriminately kicks away universal scaffolding, shouldn't that completely destroy the healthy cells right alongside the rogue megakaryocytes? Why is it selective?
That is the exact critical question the researchers had to answer. They had to prove it wasn't broadly toxic. So they started in vitro in petri dishes using healthy human CD34 positive stem cells.
Okay.
They applied revumenib and tracked the cellular development using flow cytometry. And what they found was that the drug specifically stunted the megakaryocyte progenitors.
The immature baby factories.
Yes, the MKPs. Normally, to become a giant platelet-producing factory, these progenitors have to undergo something called polyploidy.
Right, which is where they replicate their DNA over and over without actually dividing into separate cells, so they just get massive.
Exactly. Revumenib completely blocked this ploidy process. It stopped them from maturing, and it triggered apoptosis-programmed cell death specifically in those progenitors.
Okay, but how do they definitively prove it's the lack of the menin scaffold causing this, and not just, the chemical toxicity of the drug itself poisoning the petri dish?
They used CRISPR to prove it.
Ah, the genetic scissors.
Right. Instead of using the drug, they used CRISPR-Cas9 to physically snip the menin1 gene right out of the human stem cells.
Wow.
And the results were identical. Snipping the gene mirrored the exact effects of the drug.
Yeah.
It proved the effect was fundamentally on target.
And to address the selectivity question, why it doesn't kill everything else, they use single-cell RNA sequencing.
The scale of this technique is wild. They mapped the transcriptomes of over 70,000 individual cells.
70,000? That's unbelievable resolution.
It really is. They aren't just looking at the cells. They're looking at exactly which genes are actively being transcribed in real time inside every single one of those 70,000 cells.
That's crazy.
They group them into distinct neighborhoods, like common myeloid progenitors, erythroid progenitors, and the megakaryocyte progenitors.
So when they dropped the revumenib bomb into this highly mapped environment, what actually happened?
Surgical precision. The drug essentially erased the megakaryocyte progenitor clusters. But, and this answers your earlier question about toxicity, it actually allowed other cell populations, like common myeloid progenitors, to expand.
Oh, wow.
Yeah. The scaffolding loss was lethal to the rogue factories, but the other cells easily adapted.
And that RNA sequencing revealed something else pretty massive, didn't it? It showed the drug was doing all of this without suppressing the JAK-STAT signaling pathways.
Right. It left the radio signals totally intact, proving it was operating on a completely novel mechanism compared to standard drugs like ruxolitinib.
Which brings us back to the scaffolding analogy. If the scaffolding is gone and the radio signals are untouched, what specific genetic building is failing to get built? The researchers traced this down to a master switch, didn't they?
They did. The sequencing data led them to two specific target genes that completely rely on menin, MEIS1 and MEF2C.
Okay.
When menin is blocked by the drug, the expression of both these genes just plummets. But they needed to find out which one was the true linchpin, so they performed a rescue experiment.
I love the mechanics of a rescue experiment. It's basically like stress testing a system by forcing it to override the drug.
Exactly. They used a lentiviral vector to engineer human stem cells that massively overexpressed MEF2C. They just flooded the cells with the MEF2C protein and then hit them with the revumenib drug.
And because the cells were flooded with that master switch, kicking away the upstream scaffolding didn't even matter anymore? The drug couldn't kill them?
Spot on. Overexpressing MEF2C completely rescued the progenitor cells from apoptosis. This proved definitively that MEF2C is the non-negotiable master switch. Block menin, MEF2C turns off, and the megakaryocyte factory goes bankrupt.
Okay, so let's do another quick recap. Menin provides the scaffolding for gene activation. Blocking it specifically starves the megakaryocyte factories by turning off that MEF2C master switch without harming the overall cell signaling.
Beautifully put.
So think about the complex systems that you rely on every day. What is the invisible scaffolding holding it all together? And, what would happen if someone suddenly pulled it away? It's wild to think about.
It really makes you appreciate the fragility of these cellular networks.
Totally. So if I'm tracking with you, we found a way to selectively bankrupt these rogue factories by kicking away their scaffolding. But forcing a switch to flip in an isolated cell line in a lab is a long way from clearing the concrete out of an actual bone marrow cavity.
Oh, a very long way.
So how did they bridge that gap?
By moving into in vivo testing the animal trials. And I have to say, they were exceptionally rigorous here. They didn't just use one mouse model, they used 4 entirely distinct models.
Okay, let me jump in there, because 4 models almost seems like overkill. If you have one solid model of a disease, why spend the time and resources creating 3 more variations?
Well, it comes down to the reality of clinical algorithms and human genetics. PMF isn't a monolith. In the real world, one patient might have a JAK2 mutation, while the person in the next bed has an MPL mutation.
Right.
If you're claiming you have a broad therapeutic breakthrough, it cannot be a one-trick pony. The drug has to be agnostic to the specific upstream genetic trigger. It shouldn't matter how the factory mutated, the drug just needs to be able to shut it down.
It has to target the downstream consequence. Okay, that makes perfect sense. So what were these 4 models?
First, they used a JAK2 V617F retroviral transplant model. This mimics a severe form of the disease, wildly high blood counts, and heavy bone marrow fibrosis.
Yeah.
They treated these mice with revumenib for 5 weeks.
And this is where we actually see the concrete clear, right?
Yes. We grade bone marrow fibrosis using something called a reticulin grade, which scales from 0 to 3. The control mice, the untreated ones, had a thick, scarred marrow with a grade of 1.5.
Okay.
Every single mouse treated with revumenib dropped to a reticulin grade of exactly 0.
Wow. Grade 0. So it didn't just slow down the scarring, it reversed it. It cleared the concrete completely.
Their bone marrow architecture went completely back to normal, but, they didn't stop there. They tested an inducible JAK2 model, which mimics an earlier stage disease. In that one, revumenib normalized their enlarged spleens just as effectively as ruxolitinib.
And then they threw the hardest test at it, right? The MPL W515L model.
Yes. From a clinical perspective, this is a notoriously aggressive mutation. It causes massive lymphocytosis, intense myelofibrosis, and it kills very quickly.
So how did they administer the drug for this one?
They mixed it right into the mouse chow. They tested different dosages to find the sweet spot, which turned out to be a 0.1% concentration in the food.
And the result?
At that dose, liver weights normalized, fibrosis reversed, and the survival data was just staggering. Every single mouse treated with revumenib survived to the endpoint of the study.
Wait, really? Every single one?
Yes. And that endpoint was weeks after every single untreated mouse had already died.
That is incredible. And just to be comprehensive, they ran a fourth model, the MPL-S504N spontaneous knock-in, and they saw the exact same disease reversal. But what about healthy tissue? Did they test this on mice without the cancer?
They did. They treated healthy mice for 12 weeks, and it was highly tolerated. The healthy mice experienced only modest drops in blood counts.
Which proves the drug is highly selective for the hyperproliferative malignant cells, leaving the normal baseline blood production mostly alone.
Exactly.
Okay, this is already an incredible breakthrough. But the paper goes one step further into something really profound: synergy. Because ruxolitinib is the current standard of care, the researchers ask the obvious question.
Right. What if we don't throw the old drug away? What if we use them both at the same time?
Biologically, a combination therapy makes total sense here, doesn't it? Because they target completely separate vulnerabilities.
Precisely.
It's a two-front war. Ruxolitinib is the signal jammer. It cuts the radio lines so the cancer cells stop receiving the grow commands, but the physical factories are still sitting there.
Yes.
So then revumenib comes in as the demolition crew, pulling out the menin scaffolding and collapsing the factories themselves.
That's a great way to picture it. You are silencing the command and destroying the infrastructure simultaneously. And to prove this mathematically, they tested the combination on a human JAK2 mutant cell line and calculated something called a ZIP synergy score.
Okay, how does that score actually work? Is it just like adding the two effects together?
No, and that's the crucial distinction. The ZIP score, which stands for zero interaction potency, it compares the observed effect of the combination against the expected additive effect. Meaning, if drug A kills 10 cells and drug B kills 10 cells, an additive effect is 20 cells. But synergy means they amplify each other exponentially. Any ZIP score above 10 indicates formal mathematical synergy. Revumenib and ruxolitinib scored a 13.1.
Oh, wow. So they act as a massive multiplier for each other.
They do. And when they put this multiplier into the highly aggressive MPL mouse model, the results blew standard therapy out of the water. Neither drug alone could significantly reduce the mutant allele burden, the core population of cancer cells.
But together,
the combination drove it down massively. They achieved a highly significant p-value of 0.000013.
Which is huge. And it also crushed the inflammatory cytokine levels, right? That toxic sludge we talked about.
Yes. Blood plasma levels of TGF-beta, MIG, MIP1 alpha, they all plummeted. And only the combination therapy completely restored the normal, healthy architecture of the spleen.
And then as the ultimate proof of concept, they bypassed the mouse genetics entirely and used PDX models, right?
Yes, patient-derived xenografts. This is the gold standard for preclinical testing.
Because they're basically using actual human cancer cells.
Exactly. They took actual CD34 positive stem cells from a human patient suffering from PMF and engrafted them into immune-deficient mice.
So it's a human tumor growing in a living proxy.
Right. And after treating these mice, revumenib significantly eradicated the human tumor cells in both the blood and the bone marrow.
Man, it's just a phenomenal cascade of evidence. You start with this strange side effect in a clinical trial. Then you trace the mechanism down to a specific scaffolding protein, menin.
Right.
You find the master switch, MEF2C. You prove that flipping that switch clears the fibrotic concrete across 4 different genetic mouse models. And finally, you mathematically prove that combining it with standard therapy creates a synergistic two-front attack that fundamentally beats back the disease.
It really highlights the absolute necessity of open-minded observation in science. I mean, the drug was built for acute leukemia. If those researchers had just dismissed the plummeting platelets in that initial trial as a random, unfortunate bug, this entire therapeutic pathway for bone marrow cancer would still be completely undiscovered.
It really makes you rethink how we look at failures. Not just in medicine, but in general. A massive breakdown in one system might actually be the precise, perfectly engineered mechanism you need to solve a completely different problem.
Absolutely. It's a reminder that nature doesn't waste mechanisms. We just have to be clever enough to redirect them.
Which brings us to our final takeaway. Testing across multiple models proved revumenib clears bone marrow fibrosis, and combined with ruxolitinib, it creates an exponentially powerful synergy. So when solving your biggest challenges, have you ever found that combining 2 completely unrelated tools creates a synergy far more powerful than either one alone?
It's a great question to ponder.
It is, and it leaves us with one last lingering thought for you to mull over today. If a severe, dangerous side effect can be reverse engineered to cure a completely different, fatal disease, What other errors or adverse events in medical history are just waiting to be looked at through a slightly different lens? What if the cure to our worst diseases is already sitting right there on a pharmacy shelf, disguised as a side effect we've been trying so hard to avoid?
Gives you chills to think about it.
It really does. Thank you for joining us on this deep dive. Keep questioning the information around you, look closely at the anomalies, and we'll catch you on the next one.